Hello, my name is Terry Furton, and today I will be talking about real-time reverse transcription polymerase chain reaction, or real-time RT-PCR for short. Basically, this is a little overview of what I'll be going over, including what happens with those nasal swabs once we collect them, to extracting that RNA and purifying it, and then finally, what exactly is going on in the thermocycler itself. So to begin, we go ahead and collect the sample, and once that nasal swab has been collected, it gets placed into like a saline solution or a PBS, phosphate buffer saline solution. And basically what that does is it gets all those cells from that swab and puts it into the solution. And that solution doesn't degrade it or anything, which is important because you don't want to degrade it if you're storing that sample. And basically that solution will then eventually, when you are extracting DNA, get placed with lysis buffer and PK to kind of lyse the cell and get the stuff out that you don't want. Basically degrade the stuff that you don't want. Which leads me into the next slide, the extraction itself. Now, a lot of labs with how high throughput COVID testing is right now have bigger machines that kind of can do this all in one swoop, but I'll be talking about it in a spin column kit format because it's a lot easier to understand that way. So like I said, you're going to want to lyse the cells first. There's going to be an incubation period in this, and basically that period is going to go ahead and allow the cells to lyse. But once you do that, you're going to go ahead and place that sample into a spin column kit, which looks like this little spin column right here. And that gets placed into a bigger microcentrifuge tube. And then you're going to go ahead and spin it in the centrifuge. And basically the sample, anything that is not RNA, will be at the bottom of the tube, and that RNA will bind directly to the bottom of that spin column. So then you're going to go ahead and take that spin column and put it into a fresh new microcentrifuge tube. And that will lead us to our next step, which is washing. Now each kit has different washing steps, so it's hard to say how many washing steps there will be, but it's basically all the same. You're going to wash everything away that is not RNA in that binding site. And again, that RNA will stay there. But anything else that is not will end up at the bottom between each centrifuge. Then the final step is the elution step. I use elution buffer. That is really the only thing that can get that RNA to unbind from that site and be collected into the tube, which is what you want. So once you do that, you're going to centrifuge it, and you'll have your final product, which will be purified RNA. And you can test that through nanodrops. There's certain standards that tell whether you have a purified sample or not in the concentration, but that's talk for another day. So now you're going to want to go ahead and set up for your PCR, well, your RT-PCR I should say, because you were doing reverse transcription. So basically you're going to want to make a master mix, and in that master mix you're going to want to have different amounts of different things. And again, the amount is not specific, that's just based on the SOP that you're using, right? So basically, you're going to want to add your two enzymes. That is the same, the reverse transcriptase and the DNA polymerase. Then you're going to want to add your DNTPs, which will help complete the strands. Then you're going to want to add your forward and reverse primers. Those are going to be specific to the fragment that you are trying to get, so that whatever the target fragment is for COVID-19. And then finally, you're going to have your probes, your Tachman probes, but I will talk about those probes on the next slide. Once you have that all done, you're going to go ahead and add that to your 96 well plate. So let's say you don't have 96 samples, and you're not going to use, obviously, all those wells, but if you do have a bunch of samples, you're typically going to want to, like the plates mostly have like 94 samples, and then the last wells are used for controls, because you're always going to want to add controls to make sure nothing went wrong. Yeah, so you're going to go ahead and add your master mix, and then you're going to add your samples. And hopefully you have a multi-channel pipette, because doing them individually sucks. Trust me. Anyways, so now let's talk about what's actually happening in the thermocycler itself. So the first step in that process is the reverse transcription, like we talked about. You're going to want to make that RNA into cDNA. And to do that, the thermocycler does it itself, but what is happening actually in there? So basically, the primer starts the new strand of DNA, and that's when polymerase comes in and completes it. And once you have that, then you can go on to the actual real-time PCR itself, where the cycles begin to take place, which take place in basically 3 steps, which is denaturing, annealing, and then extension, right? So typically in most thermocyclers, that cycle 1 is where, I know this diagram kind of doesn't really show it, but the probe doesn't really attach in cycle 1 typically. You're going to basically be replicating that cDNA that you just made. So that's where that process will take place, and that's kind of similar to what just happened, where you're going to add those primers. The primers are going to attach to the strand. Those DNTPs are going to basically fill anywhere that they need to fill, and then polymerase is going to go ahead and come and complete that strand. And then that leads us to cycle 2, which will actually add the probe. So again, we're going to have denaturing, so the heat will be higher, which will make those strands separate. And then once they're separated, the annealing process will happen where the primers attach. But this time we'll have the probes attach. And now these TaqMan probes are basically indicators of whether or not you have something that they're targeting, right? So these probes attach specific sequences. And if they attach, you have a reporter that attaches. That reporter is basically illuminates fluorescent light that can be detected by a camera, which we'll talk about on the next slide. And then you have a quencher, which attaches at the end of it, which basically prevents that light from happening while the strand is being completed. So once that happens, extension, the last part of the cycle happens, extension. Basically, polymerase comes in, and here's kind of like a picture of it. It completes the strand and cleaves off that reporter and that quencher. And with the quencher cleaved off, that gives basically the reporter permission now to give off that fluorescent light. And that's basically what's happening in this process. And this gets repeated for most cycles, like 35 cycles. So you're getting all this DNA, which is getting basically doubled every time, amplified every time. And all these probes, if they're being attached to that specific sequence, you have these probes that are now basically emitting fluorescent lights. So finally, we have basically a graph that is made through the machine. Now a camera is able to detect that fluorescent light that is being given off. And after however many cycles the thermocycler is set for, you'll get some sort of graph that looks like this. Now this is just obviously 2 samples, but if you have more samples, there will obviously be more lines. Now, like I said, when those samples get cleaved off, they start to emit light. And the fluorescent light, we kind of have like the number of cycles here on the x-axis and the fluorescent light on the y-axis. And then we have something called your CT value. But first, let's talk about the threshold. So the threshold is basically the level of detection, or the point at which reaction reaches a fluorescent intensity above that background levels. And now the number of cycles is kind of where that increase happens, which gives us our CT value, or our cycle threshold value. Now that's important because in positive cases, you're going to want detection below, basically anything below 30 cycles is typically a positive case, which means anything, if we're seeing below 30 cycles, so like let's say at like cycle 25, we already see that increase in fluorescent lights, which means those probes are attaching to specific sequences in that DNA and then being cleaved off, and it keeps getting amplified, so it keeps happening. So that's how you have a positive case. And this is my sources.